Hello again. In lesson 7, we're going to be talking about neural cells. And neural is a word that I use to describe anything that's nervous tissue. So we're talking about different cell types within the brain, spinal cord, central, peripheral, what have you. The basic unit, the basic cell of the nervous system is the neuron. And some of this, I'm sure, is going to be review, and some of it, I'm sure, will not be review. So we're going to start off with the neuron. We're going to start out talking about the structure of the neuron. Now, the neuron is the cell in the nervous system that is responsible for signal propagation. It is responsible for communicating signals both electrically and chemically over both short distances and long distances. And it utilizes a very specific system to accomplish this goal. Here in the structure of the neuron, you can see that we have what we call a cell body, or a soma, shown here, and a nucleus. So all cells have nuclei, obviously. The nucleus essentially is the life of the cell. So it's going to contain all types of things, and we'll talk about that in a minute. Here we have, extending from the cell body in this particular neuron, we have what looks sort of like a little tree with tree branches. Now these are the dendrites. So these are processes or neurites that are extending from cells. And here on from the other part, if we come down to the base here, we have this long extension, which is what we call the axon. Okay, and the axon terminates in what we call axon terminal boutons. All right, so these are the terminals. Okay, and these are the sites of neurotransmitter storage, synthesis, etc. And this is where the neurotransmitters are going to get released. And we'll talk a little bit about that in more detail as we go. Outside of the axon here, you can see that we have this thing called a myelin sheath. So like when we go back into ancient history and we started talking about the nerves as wires analogy, and we noted that much like a wire that carries electrical current, so does the neuron carry electrical current. And one of the things that insulates this current or keeps the current functioning as efficiently as possible is this insulated layer. Right? That's provided by this myelin sheath around it. So these are some of the very basic components of the neuron, things that you've probably done before. And what we're going to do is we're going to get a little bit more detailed, and we're going to talk about how really neat these cells are and how they function in cooperation with other types of neural cells. So this is just to review. We have all the normal players in our nucleus. We have our nucleus and our nucleolus. We have a membrane. Here we have a ER. Okay, so this is the endoplasmic reticulum. We have some rough area here. We can see these ribosomes attached to it. We have mitochondria to provide ATP, which is important actually in maintaining and getting back to resting membrane potential, which we're going to talk about. And then we have these sort of spaces here called synapses. And if we were actually, there's a synapse over here, one over here, we can come around, and there's some up here too. And if we take a closer look at the synapse, what we can see is that it's essentially a marriage of neurons. Marriage of one neuron that is sending signals to a neighboring or to an adjacent neuron. And in reality, these neurons can be far away from each other or they can be closer together. It just depends on where we're looking. Right? So some other things that I wanted to note in here is here we have these things called microtubules. Right? Now you should be familiar with microtubules from back in biology. And if we take a closer look here at the axon, we see that we also have microtubules. So we have them not only in the cell body, but we have them extending all the way down the length of the axon. Right? And we have microfilaments. Now, these are super important cytoskeletal elements. They have several key functions. One function is actually to extend the axonal process. So You may recall from development that as neurons are developing and as they're sort of finding their synapses and moving around, one thing that has to happen is axons need to send their projections and they need to reach their destinations, form the synapses that they're going to form. Some of those synapses will stabilize and become permanent. Some of those synapses will degrade away and essentially are non-functional. But we do have to have axonal extension. All right, so remember, axons and dendrites collectively are referred to as neurites. So a neurite could be an axon or a dendrite. But what these things also do, right, is these microtubules provide a highway for different molecules to move up and down the length of the axon. All right, and this is particularly important for synthesis of things like proteins and neurotransmitters, etc. And they actually move on little motors that attach to them. These things you may have heard of, kinesin or dynein. These are a couple of the ones that are used. We're not going to get into too much detail about those, but there are molecular motors that attach to the microtubules, and it's a really neat sort of method of transport that can get things from point A over to point B down here. Right? Now the rest of the details of the synapse we're going to go through as well. Some different ways that neurons can be classified. There are several that I'm going to discuss with you. There are several others that I'm not going to talk about, but suffice it to say, it sort of just depends on what you're examining. So for example, I talked about neurites already. We can classify them by how many neurites they have, okay? And in this classification, we can make a distinction between a neuron type that is what we call unipolar or bipolar or multipolar. I'm going to show you some pictures of these in a minute, so just bear with me here. We can also classify them by what type or shape of dendritic tree they have. So I showed you in the last slide how they sort of form kind of a little branching pattern. Well, there's different branching patterns, and some neurons have more or less dendrites, and they tend to be shaped in different ways, depending on which structure you're looking at and it's actually related to their function as well. Remember, in neuroanatomy, just like any anatomy, there's always the structure-function relationship. So they can be classified by dendritic trees or by what we call dendritic spines. Okay? So you may hear of a pyramidal cell or a stellate cell. Now these are two different types of cells that have different arrangements of their dendritic trees. They're shaped in quite a different way. Okay, the pyramidals tend to be shaped more in a pyramid, whereas the stellates look more sort of like star-shaped. Or you might see a cell that's called spiny. Now this refers to the fact that the dendrites have protrusions emanating out of them called spines. These are dendritic spines. And what dendritic spines are clusters of super dense clusters of receptors. So they have their very specialized structures that are present in certain types of synapses in the brain, particularly. Okay? Or we can classify them based on the types of connections that they make. So we know that we have sensory neurons, which are distinct from motor neurons. And in fact, we have interneurons. That's something that you'll get a nice sort of view of that in one of the video clips that you're going to watch today. But we have these different types of connections that neurons make. Okay? And finally, for us, the last one that I'm going to discuss with you is by the type of neurotransmitter that is synthesized by the neuron. So you may have heard that, for example, Parkinson's disease is a disease that's characterized by loss of what we call dopaminergic neurons. So what does dopaminergic mean? Well, dopaminergic means that they are neurons that synthesize, drumroll, dopamine, okay? Versus cholinergic neurons, cholinergic synthesize acetylcholine, okay? So this is yet another way of classifying. So I wanted to show you some images. So here we have classification based on the neurites, okay? So here we have bipolar. So this essentially is telling us that we have two separate extensions of neurites. So even though in general there's more than two neurites here, but we can see there's two main branches of neurites that are formed here, okay? Whereas unipolar, we sort of have one, and there might be a couple of processes here, okay? This is what we know of to be a sensory neuron, so there's also a little bit of the connection type here as well. This is what we would call a multipolar neuron. So the majority of the neurons in the brain are actually multipolar. And what we can see here is we have the axon emanating out, and we have many different branching points for dendrites, okay, where we're going to have dendritic trees sprouting out from here, okay, so this is multipolar. Then we have an example of a tree-like classification. So this is actually a pyramidal cell. Now these cells are present in large quantities in the hippocampus of the brain, particularly. So you may remember the hippocampus from your neuroanatomy. That's actually this important for both short-term and long-term memory storage. And there are particular cellular layers in the hippocampus that are composed entirely of what we call pyramidal cells. And they're called this because, as you can see, this sort of looks almost like a little pyramid shape of dendritic arborization here. All right. Here's an example of connection type. So we can see here is a particular type of neuron called an interneuron. Now, neurons really are sort of bridges that are going to go from one neuron type to another. So you're going to see an example of an interneuron that actually will receive a signal from a sensory neuron and send the signal directly to a motor neuron to engage in reflex behavior. So this is something that the body does, and it's actually really neat. Here we have a motor neuron. So this is, again, a sort of multipolar morphology, right? But this is a classic motor neuron. Now, most neurons tend to have, most motor neurons tend to have this type of morphology. And here we have a sensory neuron. So the sensory neurons will begin from some sort of tissue, okay, or a cell that is a sensory region. So, for example, we have receptors in the back of our retina that receive light. We have a retinal tissue, and then we have receptors that actually will carry that signal through the optic nerve, and it's going to carry that information all the way back to the visual processing region of our brain, right? Also, here we have classifications based on spine or neurotransmitter type. So I wanted to show you this because this is a really interesting, neat part of neurobiology. This on the top here in A is sort of masked because this box is on top of it, but there was an A here for this figure. This was taken from, I believe it was the Journal of Biological Chemistry, a really elegant study that was performed using some fluorescent microscopy, which is shown here. Okay, so fluorescent labeling, and also some electron microscopy and some cellular staining. So these are really advanced techniques. And what you can see here is this is a single dendrite with a bunch of dendritic spines that are actually labeled here coming off of it. So these are very dense clusters of receptors, basically. Okay, dendritic spines have lots and lots of receptors. They're highly concentrated. Okay, this is a close-up view. Just looked sort of neat, so I included it. And here we can see on top of this is actually, excuse me, this is actually a spine here, but what this spine shows you is that it actually contains a bunch of receptors called glutamate receptors. All right. Now, the neuron that may synapse with this actual type of spine is most likely a glutamatergic neuron. Okay, so that would be characterizing it by neurotransmitter type because it would be a neuron that synthesizes glutamate. Okay. Now, how do neurons signal? These are the basic big ones. They do communicate with each other rapidly and often, and they do this by receiving and sending signals not only from other neurons but also from different cells and other kinds of tissues. So, for example, we have neuromuscular junctions. So we have a neuron that will synapse with a muscle tissue and will actually propagate an impulse, an electrical impulse throughout the muscle tissue that will cause an increase in calcium that will cause contraction of skeletal muscle. Okay, so it's not always the way that you might be thinking about it now in terms of neuron A connects to neuron B, connects to neuron C in different regions of the brain. We also have tissues of the body, such as glands and other organs, muscles that need neuronal, what we call innervation. So innervation means that the neuron is actually going to project to it and signal to it. Okay, so it innervates it. So in the neuromuscular junction, the neuron will innervate the muscle tissue. Okay. So neurotransmission is what we call propagated sometimes. That's a good word. It's used in neuroscience a lot. Propagated by electrical signals that are driven by action potentials. So the action potential occurs when the neuron has received enough stimulation and has taken in enough ions that have changed its membrane potential enough for it to actually fire a complete action potential. It's an all or none phenomenon, and I'm going to have you watch a quick video on that because it's cool and it's animated, and I think it does a pretty good job of summarizing this all up. Also save my voice a little bit. All right. Neurotransmitters are the signals, so these are the chemical portion of the signaling. So we have a two-pronged effect. We have chemical signals. So neurons release chemical signals called neurotransmitters that then bind to receptors on the postsynaptic neuron that cause that neuron to become activated or deactivated, as the case may be, right? Sometimes they're inhibitory signals, okay? Now, the binding of these neurotransmitters to the receptors, we sometimes think of as binding of a ligand to a receptor. So a ligand is a molecule that can bind specifically to a receptor. And remember, receptor-ligand interactions are very much like a lock and key sort of arrangement, meaning that if I have a key to a particular lock and I go to my friend's house, my key is probably not going to fit in that lock because my key was cut in the exact same specific shape for that lock. And we remember from our knowledge of proteins and structure that the three-dimensional proteins of structures of proteins rather are critical in their function. And that three-dimensional structure is going to determine whether or not it has ligand binding capacity for a certain receptor. Okay? And so this type of transmission is actually unique to neurons, although there have been more and more studies that have documented that astrocytes actually can do some signaling that is somewhat similar to this, but this is typically thought of really as a neuronal phenomenon, okay? So, yes, they form synapses. Here's a presynaptic cell. So we call this the presynaptic neuron because it is the one before the synapse. Here would be our synapse. So this is actually the terminal buttons of or boutons of the presynaptic cell synapsing with portions of the postsynaptic cell here. Okay, so we have the terminals and we have dendrites, and we may even have dendritic spines over here, depending on where we are, right? What kind of area this is. So here we have the signal, which is going to travel presynaptic to postsynaptic. We're going to have a release of chemical stimuli, right? So we're going to have neurotransmitter release that's going to bind to receptors and ultimately either going to help to trigger an action potential or help to diminish the probability of triggering an action potential. And it just depends on whether or not we're talking about an inhibitory or an excitatory neurotransmitter. All right. So at this point, there's a couple of really neat videos that I would like you to watch. I have the links here, so I'm going to let you guys do this rather than just playing them on here so you can get them. You can also get the links from the PowerPoint that I'm going to put onto OneDrive if you're having trouble typing it in or something like that. But this is going to show you a really nice summary of synaptic transmission, and I think they use dopamine as an example in this one, but it could be anything, okay? And then the second video is actually on the generation of an action potential. Now, action potentials involve a summation of inputs, right? So an action potential can only occur when we've reached a certain membrane potential, all right, which we call the threshold potential. And it has to do with the types of ions that are coming in and leaving, and it has to do with not only the types of ions, but the gradient that's established. So this is stuff that I think you guys have had before, so I'm not going to spend too much time going over it. Things that you should know and be comfortable with are listed here, okay? So you can go through this list, see if you understand these different parts. And I'm going to go to the next part, which is my quick overview. All right. So a neuron at any given point, if it is at rest, probably has a resting potential of about negative 70 millivolts, give or take a little bit. You may see negative 65, you may see negative 72. It's roughly about negative 70. We're going to call it negative 70. Okay. Now that is the resting potential. Okay. In order for this neuron to fire, what has to happen is that potential has to change. And that potential has to change by going up, okay, to somewhere in the neighborhood of about negative 55 millivolts. Okay. So we need to have a change based on some stimulus. Now, what is the stimulus? The stimulus for any cell are actually the binding of chemical stimuli to receptors. And the way that we change a membrane potential is actually by changing the types of ions and the concentrations of ions, right? So we've got these different channels in the membrane of the neuron, okay? These receptors, many types of receptors form channels, and we're going to go over this in a little bit more detail when we go through ionotropic and metabotropic channels and things like that, right? Different types of receptors. Some of them have channels, some of them don't. Some are conformational change. But the idea here, all right, is that different neurotransmitter binding to receptors is going to result in either directly or indirectly the entrance of ions into the cell or the exit of ions out of the cell. Okay, that's going to change our potential because we have ions like sodium and calcium, which are positively charged. Okay, potassium's another one, and we have chlorine, which is a big one that's a negatively charged ion. Okay, now depending on which way these things move, it's going to either increase or decrease, right, how negative or positive the potential is. So when enough signals have entered a cell, okay, that's caused enough shift in the ionic balance where it's gone to about negative 55 from negative 70, it is going to do something that we call depolarizing, all right? It's going to depolarize and it's going to initiate an action potential. And what that means is that we're going to start taking in sodium, rapidly taking in sodium. It's going to go more positive, and this is going to happen like a chain reaction all the way down the axon, over and over and over again. Sodium entry, sodium entry, sodium entry, and it's going to go real fast down the axon till it gets to the end. When it gets to the end, it's going to trigger the release of whatever neurotransmitters are made by that cell. Okay, and that involves calcium, which is something a little bit separate. Okay, so that's a little bit separate from the action potential. There's actually two phases. Okay, there's a calcium component at the end. All right. Some neurons actually do rely on calcium quite heavily to depolarize. It just depends on which type you're talking about. All right, so it could be sodium, it could be calcium, it could be both. All right, and then once this has happened, the membrane needs to basically reset and it needs to repolarize. And so what happens during repolarization, in large part, okay, is that potassium ions go out. And we keep kicking out these potassium ions and getting rid of the positive charges so that we can make the membrane potential more negative, okay? And ultimately, at the end, we need the help of an ATP-driven pump, which I think you guys have had this already as well, the sodium potassium ATPase, which actually will restore the resting membrane potential and keep it where it needs to be, okay? These are important mechanisms, both the repolarization and the refractory period here, because all of these rectifying channels and these potassium channels actually help to ensure that the cell, number one, doesn't just continue to fire over and over and over again, and number two, that the current doesn't actually go back the other way. Okay, so this is interesting mechanism. I'm not going to get too much into the ions in here other than the fact that you should know the basic major components of the action potential. So you should understand what happens with the sodium and the potassium, okay, and you should understand what happens with the ATPase. So if you know this diagram and you know something about the ions involved, you should be in pretty good shape, okay? Now, enough about neurons, because I'm really a glia guy myself, and I love glia because everybody talks about neurons, and for so long, I mean, decades and decades, glia were largely ignored. Up until probably, maybe about maybe 20 years ago, something like that, at least in fields of neurodegenerative disease. And that's sort of my expertise, right? Because everybody just sort of thought, well, you have all these neurons, they're important, they're sending all the signals, they're doing all the work, and these glia are just sort of there. You know, they're the glia. What is it? It translates actually into the glue. They're the glue. They're just sort of there in between the spaces, maybe providing a couple of nutrients here and there. But it turns out that it's much different, and they're a lot more important than that, and they do lots of amazing things. Okay? And one of the things that I've actually spent a long time studying is the reactions of certain type of glia to injury, okay, and treatment as well. So these are critical in the function of both the central nervous and the peripheral nervous systems. All right. Now there's different ones. You can see here from the schematic that the CNS, we have these four major types, whereas the peripheral system, uses a different supportive cell, which we call a satellite cell, in contrast to the astrocyte of the central nervous system, and it uses a different myelinating cell called the Schwann cell, which is in contrast to the oligodendrocyte, which is really the myelinating cell of the CNS. All right? But in either case, essentially, the same functions are performed in both, but we do need more specialized function than the central nervous system by virtue of the protection that it needs to enjoy, and also the fact that it needs to respond to pathogens and injury a little bit differently than the peripheral nervous system due to something called the blood-brain barrier, and we're going to talk about that later too. Okay, so we got a couple more players in the CNS, and they're pretty cool ones, but really all of these cells work together and they form a network and they all support each other and their functions are connected. You really can't think about the nervous system without thinking about glia, and certainly not the central nervous system. So here I have a schematic that depicts neurons in what we call neuroglial cells. All right. So typically when you hear or you see the word neuroglial, people are talking about the glial cells of the brain. So here we have a neuron. We can see the neuron here. There's a microglia alongside of it. Now we don't really know what it's doing yet, okay, but we'll get there. Here we have an astrocyte, and not only is this astrocyte up next to the neuron, because it's actually going to be providing things that the neuron needs, but it's also butted up against something else that's not present in this diagram. And we're going to see that later, which is a blood vessel. So these glial cells actually provide important roles to vessels and preserving the barrier in the brain as well. And here we see the oligodendrocyte. This is its cell body, and it's actually extending branches that's allowing it to myelinate a couple of different neurons. So we have one neuron over here, we have another neuron on the other side. Okay. Now, one of the cool things about astrocytes in particular, okay, is that they do more than one thing that's very important. First of all, yes, they do provide some nourishment. They do buffer the extracellular fluid. So one thing that we have to maintain at all costs is the pH and the appropriate ion concentrations of the different ions that are contained within the extracellular fluid. And astrocytes are super important in this capacity. So they do tend to regulate things like, for example, the extracellular potassium concentration. So from your video that you just watched on the action potential, you should have learned that potassium is super important, and keeping a high potassium concentration inside the cell is a very important thing to do. Okay, so we always have to make sure that we're maintaining the right concentrations. In addition to that, though, the astrocytes also help neurons to mature and differentiate and then be able to produce the right types of molecules, neurotransmitters, proteins at the right times. And they do this by producing things called growth factors. So every neuron, it doesn't matter what neuron it is, relies very heavily on access to certain growth factors to be able to perform all of its functions. And a lot of these are actually made by astrocytes. The other thing that astrocytes do is they sequester the neurotransmitter glutamate. Now, we haven't done neurotransmitters yet. We're going to get into that very shortly, actually. Not in this lesson, but probably in the next one for sure. Glutamate is the major excitatory neurotransmitter of the brain. And GABA is the major inhibitory neurotransmitter of the brain. So by and large, the brain uses GABA and glutamate to signal. Now it uses other ones too that are super important, but not in such large quantities. So there tends to be a lot of glutamate around. And one thing that glutamate can do is it can induce toxicity of neurons. Now you might say to yourself, well, why would neurons make something that's going to kill them? They do. They actually make lots of things that are going to kill them, all right? But what happens is that regulating the balance appropriately prevents this from happening. And one of the players that does this is the astrocyte. And they actually will sequester glutamate and make sure that too much glutamate isn't present that actually becomes cytotoxic to neurons. So that's super important. We talked about the oligodendrocytes already. I just wanted to add a couple of things about this that we're going to get to later. Well, the one part, it's just something interesting to note. We did talk about how the Schwann cell is the myelinating cell of the peripheral nervous system. Well, oligodendrocytes are important in a major way, and that is that they can actually myelinate multiple axons. And it's been observed that a given oligodendrocyte can myelinate probably upwards of 50 different axons at once, okay? Whereas the Schwann cell is really just a one-trick pony. The Schwann cell is going to grab onto one axon and myelinate it, and that's it, okay? So the oligodendrocytes are a lot more dynamic. They can do a lot more for you. Now, in certain types of disease states and certain types of illnesses that occur even actually from the point of early on in development, childbirth actually, what's been recognized about oligodendrocytes is that they develop in stages and they differentiate into, they start as a pre-oligodendrocyte, and then they become an immature oligodendrocyte, then a mature myelinating oligodendrocyte. So they actually go through these maturational stages throughout development. And the immature population of cells is very, very susceptible to things like hypoxia or ischemia. Now, hypoxia simply means that the tissue is not getting the amount of oxygen that it should be. Okay, so it's a low oxygen condition. You can see hypo, right? Hypoxia, below, okay? So less oxygen than it should be getting. Ischemia means you're not getting as much blood to the tissue. So if you have ischemic conditions, you're not getting as much blood flow, and what does blood do? Well, it carries the oxygen, okay? So these things kind of go hand in hand. So these immature cells are very susceptible to it. So an insult early in development where you're cutting off oxygen or blood flow can have profound effects on the way that myelination occurs in the system. Because when these pre-immature oligodendrocytes are lost, it actually affects the way that the neurons function. So this is something that's become super important. People didn't know about this probably 30 years ago, but we know now. And so this is really a hot area of research. We also have these microglia. Now, the microglia are my absolute favorites in the brain. These are the resident immune cells of the brain. I say resident because it means the actual place we're in, right? You're a resident of your own neighborhood. That's the neighborhood you live in. So resident immune cells, when we're talking about the brain, are immune cells that originate in the brain. Because all the other immune cells in the body are actually floating around in the peripheral system. They're floating around in your bloodstream, and most of them never actually see their way into the brain because of this barrier called the blood-brain barrier. And we're going to talk about that again. But the microglia, because we have a limited immune capacity in the brain, because of this blood-brain barrier, and it restricts immune cells from coming into the brain has to have a mechanism, okay, to remove things like cellular debris, okay, or to alert other cells when it looks like there's something wrong. They release these chemical signals, and they produce, just like if you cut yourself and if you cut your arm, for example, and your arm will become inflamed, your body will mount an inflammatory response from that cut, right? And it's going to get yellow, and it's going to get puffy and red, and it's going to hurt a little bit. It's because your immune cells are actually producing all these chemicals that are telling your other immune cells to come and fight this off, fight the infection off and heal the wound. Well, the microglia in your brain do similar things by mounting what we call a neuroinflammatory response. So that's an inflammatory type response in the brain when something is wrong. Okay? Now, the something wrong could be an injury to the head. It could be a degenerative disease. Okay? It could be some kind of progressive brain disorder. Could be lots of things, right? But the microglia do this. They eat up, they phagocytose debris, things around that aren't supposed to be there, okay? And if there is some kind of pathogen or some kind of foreign body that does happen to penetrate into the brain, because it does happen, even though we try not to let it happen, it does happen, they act as what we call antigen-presenting cells. And what this means is that they essentially eat the pathogen, and then they rip a piece off of it, and they stick it on their membrane. And they show it to other immune cells in the body. Now, this is like a complicated sort of mechanism that your immune system uses, but it's super cool. And so these guys will act as antigen presenters because they take a piece of the pathogen that they've eaten and they present it on the edge of their cell surface so that the other immune cells can see it and start mounting a response against it. So pretty neat stuff. And the last part, and actually I don't even think this one's on here, is the ependymal cell. And these are those cells, they're super important because they actually line the ventricles, okay? And not just the ventricles, they're also in the spinal cord, because remember we have cerebral spinal fluid that goes through the ventricles, the lateral ventricles, the third ventricle, the cerebral aqueduct, the fourth ventricle, and actually down the entire the central canal of the spinal cord. Okay, so we need this everywhere in the CNS. All right, and these cells produce the cerebral spinal fluid, the CSF, and they actually this tissue, okay, that lines the ventricles, this ependymal tissue actually interfaces with capillaries, with blood vessels, okay, to form the choroid plexus, and that's actually the location, the actual seat of where the CSF is made. Right. So what is this blood-brain barrier that I keep talking about? I keep talking about the blood-brain barrier. So here you can see that we have a whole layer of cells, okay? This is what we call an endothelium, okay, a vascular endothelium, because these are a certain type of cells. They're called endothelial cells, all right? And they are the cells that line the blood vessels. We also have vascular smooth muscle. So this smooth muscle allows vessels to expand or contract. So you may know, for example, that in response to differences in blood pressure, or if you get hot, if you're working out, your vessels will sometimes dilate to push more blood towards an area, or they'll constrict to cut off the blood flow to an area. All right, and this is how they do it, is through these muscles. So we have this vascular endothelium here. This is the blood. These are the red blood cells that are floating around inside, and some other things too. I'm not sure if that's supposed to be a protein or some sort of maybe a platelet, because it looks like it's shaped a little bit differently. But in either case, okay, these are the cells in the blood. So these are definitely red blood cells. All right, and what we can see here is in the brain, so this section is actually the side of the brain. So this would be like a vessel going right along the surface of the pia mater. Now, you remember we talked about how there's these 3 different layers of the meninges, okay? And we have this pia mater, and we have this blood vessel on it, and we have a brain. And here what you can see is there are actually processes being extended from astrocytes in something called a pericyte. Now, a pericyte is another glial type of cell that actually is involved exclusively in forming connections with vessels, blood vessels that are entering the brain. So pericytes, as far as we know, and we're still learning a lot more about these cells now, it's not one of the ones that we're too familiar with their function, so it's actually very exciting as well. But these pericytes, from what we understand about them, their function is to provide nutrients and support for blood vessels, okay? Cerebral blood vessels, so the ones that are going into the brain, all right? And here we can see that the blood-brain barrier really is a combination, okay? It's a combination of the tightly compacted endothelial cells of the blood vessel and also the neighboring pericytes and astrocytes that are inside the brain. Then they all form connections here. And what they do is they restrict the entry of different types of pathogens, right? Microorganisms. They also help to ensure that toxic substances that you might get exposed to, right? So maybe you might breathe something in the air, and maybe this chemical gets diffuses into your blood, right, through your lungs and into your blood, and it's going through your peripheral system. It's going through your blood, but you don't want it to get in your brain. Because the brain, of course, is a super important organ that controls everything. And we want to make sure that it does not get exposed to things like microbes and toxins and things like that. So this very tight barrier is in place just for that reason. It also helps to maintain ionic balance, okay? So even though sometimes we can have ions get out of balance in our bodies, so for example, you might have high sodium, okay, or you might have like potassium concentrations that are off. Um, as you get older, you'll probably go to have a blood test at some point, and they're going to look at your levels of all these different what they call electrolytes, right? Well, these are the different ions that are typically present in your body, and they need to be at certain levels for things to function properly. So, point being, even if some of these levels get out of whack in the peripheral system, this can affect things like muscular function, okay? So that's one of the reasons why ionic imbalances can cause heart problems, because your heart's really a giant muscle, right? But having this blood-brain barrier here actually sort of protects your brain a little bit more from problems that you might have in the periphery becoming problems that you have centrally, okay? So this is an important thing. So here you can just kind of see that there's a neuron here. We have a blood vessel, and this is an astrocyte butting up against the blood vessel here. The pericyte's missing from this diagram, but you would actually have a bunch of pericytes here too. And what they really do is they provide nourishment and support for this, okay? So super important stuff. But now you know there's going to be a but here, right? It is great protection, but sometimes it can actually be a double-edged sword. So when we're talking about injury and we're talking about disease states, and if we're talking about brain injury and brain disease states, oftentimes we need to resort to trying to find some sort of a therapeutic remedy that involves drugs, okay? Prescribing a drug or some kind of what we call pharmacological agent. So something that we can give you that's going to bind to a receptor, change some things around in your brain, and make you feel better, right? One of the problems with this is that your blood-brain barrier is so good at keeping things out that it also keeps out a lot of drugs, okay? So when researchers and clinicians are looking for ways to try to improve patient outcomes, okay, for patients that are coming in with certain types of disorders or ailments or injuries, one of the things that they often do is try to identify an area that they think, like for example, a process, let's say that we've determined that Alzheimer's disease, if we can get rid of a certain type of A beta, okay, we can improve the outcome. Well, that sounds great, but what do we have to do? We have to give some kind of a drug that's going to affect maybe a receptor, okay, that's going to break this down. This isn't exactly the way that it works, but it's a good analogy, all right? So we need to give drug A to decrease the amount of A beta, but drug A has to get into the brain first, okay? It's a challenge. So what you then have to have are chemists and pharmacologists, different types of scientists, that are going to sit around and figure out how to make a molecule that's not only going to have the right conformational structure to bind to the target that it needs to bind to, and only that target, right? Because we don't want it sticking all over the place. That's what we call side effects, okay? We don't want this process to get improved, but at the same time, somebody to have a brain hemorrhage, okay? So you want it only to bind to your target. So not only do they have to figure out how to make the molecule that binds to the target, but they also have to figure out how to make a molecule that binds to the target, but that will also be able to pass through the blood-brain barrier. And this is not an easy task, okay? So this slide here, I just wanted to introduce this for now, but we're going to be getting back to this idea later when we actually talk about pharmacology in more depth, okay? So for right now, I just wanted to introduce blood-brain barrier, give you an idea of why it's important, okay? So you know basically what it is, and then we're going to do a lot more with this later, all right? So that's it for neural cells. I put a quiz on here that you can check out, see how you did with this one, and I will see you next time. Thanks.